So for number 2, about the nucleic acids, a nucleotide is composed of 3 parts, which is phosphate, 5-carbon sugar, and nitrogenous base. The phosphate group is attached to the 5-carbon. Of sugar, and the nitrogenous base is attached to the 1-carbon of sugar. DNA and RNA have different molecular structures and functions in the body. The key differences in their structures are sugars and nitrogenous base. With sugars, DNA contains deoxyribose, while RNA contains ribose. The difference lies in the presence of the hydroxyl group on the 2-carbon of the ribose, which is replaced by a hydrogen in an atom in a deoxyribose. And in the nitrogenous bases, DNA has 4 bases: adenine, cytosine, guanine, and thymine. RNA has the same base except for the uracil, which replaces thymine. In terms of function, DNA is a primary genetic material in cells and serves as a blueprint for the synthesis of RNA and proteins. RNA has several roles, including serving as a template of protein synthesis, messenger RNA, being part of the ribosome structure, ribosomal RNA, and assisting the process of protein synthesis, transfer RNA. Some RNA molecules also have regulatory functions. Viruses are small infectious agents containing genetic material, nucleic acid that can be either DNA and RNA. A virus consists of nucleic acid core surrounded by a protein coat called a capsid. Some viruses have an additional lipid envelope derived from the host of the cell membrane, which contains protein spikes that facilitate the entry of the virus into the host. When a virus infects a host cell, it attaches to specific receptors on the cell surface using its protein spikes. The viral nucleic acid is then released into the cell, where it hijacks the host cell machinery to replicate its genetic material and produce viral proteins. So the explanation: the newly formed nucleic acid and viral proteins assemble into new viral particles within the host cell. In the case of an envelope viruses, budding occurs where the new viral particles acquire their envelopes as they exit the host cell. So the final answer: the release of the new viruses leads to the death of or dysfunction of the host cell, allowing the viruses to infect other cells and spread infection. So for number 3, the following about metabolism. Catabolism and anabolism are 2 types of metabolic processes that occur within the cells of the living organism. Catabolism refers to the process of breaking down complex molecules into simpler ones, releasing energy in the process. This energy can be used by the cell to perform various functions such as growth, repair, and movement. Anabolism, on the other hand, refers to the process of building complex molecules from the simpler ones, requiring energy in the process. Anabolic reactions typically involve the synthesis of larger molecules from smaller ones, such as synthesis of proteins from amino acids. 2 types of chemical reactions that tend to be found in catabolic pathways: hydrolysis and oxidation. Hydrolysis is the reaction of a molecule with water leading to the breaking of a covalent bond in the molecule. So, for example, in the breakdown of glucose through glycolysis, the glucose molecules hydrolyzed into 2 pyruvate molecules, releasing energy in the process. Oxidation, on the other hand, is the loss of electrons by a molecule or atom. This is often accompanied by the release of energy in the form of ATP. So, for example, in the electron transport chain of a cellular respiration, the oxidation NADH and FADH2 releases energy that is used to produce ATP. 2 types of chemical reactions that tend to be found in anabolic pathways are dehydration synthesis and reduction. Dehydration synthesis is a process of joining 2 molecules together by removing a water molecule. So, for example, in the synthesis of proteins, amino acids are joined together by dehydration synthesis to form peptide bonds. Reduction, on the other hand, is the gain of electrons by a molecule or atom. This requires energy, which is often provided by ATP. So, for example, the synthesis of glucose through glucogenesis, ATP is used to reduce pyruvate to glucose. Adenosis triphosphate, ATP, is often called the energy currency of the cell because it is used to store and transfer energy within cells. ATP is hydrolyzed to adenosine disphosphate, ADP, in various cellular processes. The chemical equation for the hydrolysis in the ATP to form ADP is this format. In this reaction, ATP is broken down into the ADP by the addition of water molecules. This releases energy that can be used by the cell to perform various functions. ADP comparatively lower in the energy than the ATP because it has one less phosphate group. The reaction is exergonic, meaning that energy is released when the phosphate group is removed from the ATP to form ADP. So the digestion is the process by which large food molecules are broken down into smaller molecules that can be absorbed used by the body. Starchy meals are mainly composed of polysaccharides, which are long chains of glucose molecules. Digestion of these molecules starts in the mouth with the enzyme amylase. Amylase breaks down into the polysaccharides into smaller glucose molecules. The saliva in the mouth contains amylase, which starts the hydrolysis of the alpha-1, 4 glycosidic bonds in the polysaccharides. The food then travels into the small intestine, where it is further broken down by other enzymes such as maltase, sucrase, and lactase. These enzymes break down disaccharides, maltose, sucrose, and lactose into their constituent monosaccharides. Body and transported to cells where they can be used as fuel. The glucose that is absorbed by the body is used in various metabolic processes such as cellular respiration, where it is oxidized to produce ADP. Glucose can also be stored as glycogen in the liver molecules, or converted to fat for long-term energy storage. Overall, digestion of starchy meals involves the hydrolysis of complex polysaccharides into smaller monosaccharides that can be absorbed and used by the body for energy or stored for later use.